taking time out to come for this lecture. Before I introduce our speaker, I'd like to start by sharing the land acknowledgement. The Field Museum acknowledges that it was built on the traditional homelands of the Council of the Three Fires, the Potawatomi, Odawa, and Ojibwe nations, as well as the Ho-Chunk, Meskwaki, Shiok, and Miami nations. The museum recognizes that the region we now call was the traditional homeland of many indigenous nations and remains home to the diverse native peoples today. The land we walk was and remains native land. Dr. David Blackburn is the curator of amphibians and reptiles at the Florida Museum of Natural History, University of Florida. He also serves as the chair of the Department of Natural History and also as Associate Director of Research and Collections at the university. Before his current roles, Dave was curator of amphibians and reptiles at the California Academy of Sciences in San Francisco. Dave is one of the most influential herpetologists today, and I am truly honored to welcome him to be our speaker today. Dave's list of accomplishments is very long, but I'll spotlight just one for his outstanding scientific contribution to understanding the evolution and diversity of frogs and his leadership in making museum specimens widely available to researchers and the public. He was elected a 2023 Fellow of the American Association of the Advancement of Science, the world's largest multidisciplinary scientific society. Dave grew up in the western suburbs of Chicago and was an undergrad at the University of Chicago, where he once attended a talk by another amphibian loving David. We have a lot of Davids in herpetology. So he attended a talk by another amphibian-loving David about salamanders, and apparently that was all it took to get Dave hooked. Today, Dave is here to spread about the good word about frogs. Thank you, Dave, for accepting our invitation. The floor is yours. We are ready for your total frog extravaganza.
I told you only to say the last part. That was about growing up in Chicago. So I came to the Field Museum for the first time when I was 13, when my mom and I moved to the west suburbs. This is the first big natural history museum I'd ever been to. The Houston Museum, and if you've ever been there, is much smaller, sort of science center type museum. So I'm still awed by the sort of just largeness of this museum and how impressive it is. And I did have a great moment of terror as a herpetologist of being briefly lost in the ornithology exhibits earlier today. So I appreciate just how big it still is. All right, so thanks for coming. We're going to talk about frogs. So behold, the frog. I've been studying frogs now for more than half my life. I gave a talk recently at the University of Chicago. So I'm trying to give a different talk today. And so instead of talking about paleontology and morphological evolution, I thought what I'd do today is to tell you some about our work related to Africa over the past 20 years, and tell you a little bit about frogs, a little bit about Africa, and trying to give you a sense of what we've done to make sense of the timescales of frog evolution diversification on the African continent. And then just for fun, because it's weird, I'm going to tell you a little bit about a very strange trait in my favorite group of African frogs. All right, so there's a lot of frogs. Very soon we're going to pass 8,000 species of frogs. As a global community, we describe usually 2 to 3 new species of frogs every week. So we're just about to hit 8,000 species of frogs, right? And one of the things that's kind of amazing about frogs is that frogs have been on Earth looking like frogs for a very long time. So, sorry, it's a little small on the screen, but this is a time-calibrated molecular phylogeny just to make a few points. One is that the common ancestor of living frogs, right, not just of what the fossil record tells us about frog, but the crown group frog is at least 200 million years old, right? So there's been frogs through pretty much every phase of amniote evolution. There have been frogs that look like frogs, right? And you could put any of us on Earth for the past 150 to 200 million years. Anywhere on Earth, if we saw a frog, anyone in this room would be able to identify a frog because they've had this basic body form for a very long time. The other thing that's kind of neat is because frogs have been around for a long time, some, at least deep parts of their evolutionary history, the diversification reflects the breakup of major continents and supercontinents, right? So you can see that sort of this early divide between the sort of, quote, we'll call them, I don't want to call them primitive, early diverging frogs here that are alive today are all found in Laurasia, so the northern continents, whereas this sort of other massive group of frogs are Gondwanan. And beginning in the early Cretaceous, there is a divide between those frogs that are found in South America and Africa, and that stamp on frog evolution remains today, right? So a large chunk of the diversification of living frogs around the world can sort of be broadly kind of put into these bins that relate to the breakup of Pangaea and Gondwana. One of the things that we found in this paper that I think is exciting, at least from thinking about the fossil record for frogs, is that it seemed for a long time frogs have just been thought of as survivors, right? There's always been frogs, like major extinction events happen and there's still frogs afterwards. So maybe extinction events weren't really that important for amphibian biology. This was thought for frogs. It was also thought for salamanders. Frankly, nobody really thought too deeply about caecilian diversification in this because there's not that many caecilian biologists. But for frogs and salamanders, it had always been thought, well, they've just survived. Now that we have these time-calibrated molecular phylogenies, there's pieces of this that become more interesting. And one of those is that it seems like a lot of the large groups of frogs here represented by the things that are basically hyloids, like the tree frogs of North America, the water frogs, so things rana, like bullfrogs, as well as microhylids. And in North America, we just have Gastrophini. Those three big groups all seem to have originated and diversified right at the time of the extinction of the dinosaurs. It's really interesting. You know, depending on how you do these analyses, the time scales shift a little bit. But as someone interested in the fossil record, this is actually quite useful because it sets up some predictions about what we might find in the fossil record. So it seems like even though frogs are very old, a lot of the sort of diversification and groups that are important today happened at the Cretaceous boundary or more recently, which is kind of neat. The other thing, just being in North America, that I think is kind of neat about this is to show that a lot of the North American frogs that we have are all colonists from somewhere else. So even though we have a fossil record in North America going back into the Jurassic and even the Triassic of frogs in North America, those are not actually connected phylogenetically to the frogs that we have here today. They're actually very different groups of frogs. So there's been some turnover in North America over the last 200 million years. It seems probably related to the end Cretaceous, where there were frogs here, and they've been replaced by frogs that have come from elsewhere in the world. Right? So bullfrogs, for instance, colonized, that group colonized North America via Beringia probably 30, 40 million years ago. The other thing that's kind of neat about frogs is all frogs look like frogs. And Bob, who I always have in my talks, but it feels very appropriate to get to have Bob in my talk today, famously once said that a frog is a frog is a frog, paraphrasing Gertrude Stein, right? You saw one frog, you've seen every frog, right? Because frogs really do have this basic body form, right? They're instantly identifiable from their skeletons. There's really no other vertebrate that quite looks like a frog, right? They have all these weird, unique skeletal traits. Those make them useful for when we're looking in the fossil record, right? It's easy to identify some of these bones in the fossil record. But one of the things that's also kind of interesting is that from this same body form, there's a lot of variance of this body form. I feel like Fieldiana is never cited in talks, but I always have this figure in talks from a paper that Sharon Emerson wrote in the 80s. You know, there are these axes of frog diversity, right? We have very different types of frogs, things that are dedicated to living in the water, like African clawed frogs or burrowing frogs, tree frogs. And the cool part is that these sort of connections, that these ecomorphs, if you will, these connections between ecology and locomotion and body form, that these evolve again and again and again in frogs, right? So these frogs all grossly look kind of similar to one another. And every one of these pairs shared a common ancestor in the Jurassic, right? And so this convergent evolution of body forms just keeps happening again and again in different parts of the world. And so this process of convergent evolution has really been used a lot in frog biology for trying to understand, you know, why do we see the diversity? Why do we see the patterns of body forms that we see out there in the world? Having so much homoplasy, so much convergent evolution really is a useful tool for exploring those ideas. And though that's true for adult frogs, it's also true for tadpoles. I'm not going to talk really any other bit about tadpoles in this talk, but I think if you don't know this about frogs, it's worth knowing. is that there's actually far more types of tadpoles than there are types of adult frogs, right? We have lots and lots of weird microhabitat specialists of tadpoles. You can't, unless you know the phylogeny, you can't pick up an adult frog and from its body form predict what its tadpole might be and vice versa, right? So there's effectively, it's been thought of as this sort of these two different life stages evolving semi-independently from one another, right? And that's been an interesting dilemma for amphibian biologists for the last 30, 50 years to think about. So cool things to do if you're interested in larval evolution. This also at a really cool time, I think, to be a frog biologist. We are really still in an age of discovery. So as just globally for amphibians, we describe 3 to 5 new species a week. Most of those are frogs every week. We're about to hit 9,000 species of amphibians. I'm 46. I was born in 1979, and we're about to have 3 times as many amphibians as there were when I was born, which I think is crazy, right? Now, these are just names. They're not just names on a list, right? We kind of tend to think about describing new species. Like, okay, well, here's yet another one of these tree frogs, yet another one of these little brown frogs. But it's exciting time because we're doing more than that. So I'll just give one example. This is not from my work. But it is an African example. This is from Mark Oliver Rudel's group in Berlin. So this is a group of frogs that when I was a graduate student was called Petropodetes natata. Now with molecular phylogenies, we realize it has nothing to do with that genus. In fact, it's a whole other distinct lineage of frogs that's been called its own family, the Odontopetrachidae. As Mark Oliver's group worked in this in the countries of Liberia, Guinea, Sierra Leone, Cote d'Ivoire, realized that in fact it was not one species, there were like multiple species, and currently there's five currently described species of Odontopetraca. So these are, adding to that global tally, but these are cool for other reasons. So in working intensively on this group of frogs, they discovered other things that are cool as a frog biologist to discover, right? So one of them is that this group of frogs, for still unexplained reasons, although there's some unpublished data with one possible explanation, these adult frogs eat a lot of leaves. Not just any leaves, leaves of only one genus of plant. And if there's a botanist in the room who wants to ask me the genus, I don't know it off the top of my head. But they have gone through all the potential things to say like, well, are they really eating these on purpose? It seems like they are actually intentionally eating this. And I can talk more with you after if you're interested in how they figure that out. But these frogs all seem to be eating lots of this plant. It's potentially related to them, their parasite loads. So potentially this is effectively a self-medication is what it's thought to basically be, although that's not published yet. The other thing that was discovered in this group of frogs, they have weird things in their mouths. And so there's now a whole research program, this is from Marvin Schaefer, actually finding that different species have species-specific chemical compounds coming out of the glands in their mouths, right? And a lot of urine communication biology has always been about sound, right? It's the thing that's most accessible to us as humans, is we can hear the frogs. We can't smell the frogs in the same way that frogs do. But now this is yet another study system in amphibians of trying to understand chemical communication in frogs. So I like to highlight this example because it's easy just to see new species as yet another name on a list, but we're at the same time discovering cool things about what it means to be a frog as well. So frog biology, just to end on this bit about frogs, has been important to us for a long time. There's more than a dozen Nobel Prizes that have been based on frogs, right? A lot of these are African clawed frogs, but not all of them. The very first cloned vertebrate was Rhinopipians. The first tetrapod born in space, I used to say vertebrate, but then I discovered that there were some fish that predated that. The first tetrapod born in space was an African clawed frog, so they colonize everywhere, including space. And there's been more than double these Nobel Prizes on frogs. Some of them are really interesting. A lot of them, a lot of developmental biology was done on Xenopus levis. But there's also frogs. There's not yet a Nobel Prize on this, but there's really interesting biomedical research on Rana sylvatica, the wood frog, right, which can do this cool trick of effectively freezing itself near solid, stopping its heartbeat, thawing, and then coming back to life, right, which is an amazing trick. And so there's some really interesting biomedical research on those frogs. Now, that's not the type of frog science that I do. A lot of my work over the last 20 years has been based in Central Africa. Doing field biology that I know quite a few of you do. When we do this work, we spend a lot of time out at night. So for frog biologists, the night is where where our happy spot is. This is a swamp in the border between Uganda and Democratic Republic of Congo. We're mostly looking for puddle frogs in the footprints of elephants. So we work all night and then we work all day prepping specimens. And part of the goal of that for me has been to try to understand African biodiversity on a very big scale, right? So I first, 25 years ago, thanks for coming to my talk. 25 years ago, I was with this guy in the middle of the Sahara. So that was my first instance of being connected to African biology in one way or another. And I got really hooked about working on the living species in Africa, in part because it was an interesting challenge, right? Africa is an enormous place. We sometimes can forget that. There's more than 50 countries in Africa. Africa is about the surface area of the moon. So it's a very big landscape to think about biogeography, diversification, evolution. And it's also fraught with tons of problems, right? There's a lot of countries, they all have different colonial histories. So the specimens are in many different places around the world. Trying to do systematics on a group like that, challenging, right? And that's true for basically any group of organisms spread across Africa. It's also a really important time to do it. Africa is full of these amazing, rich biodiversity hotspots scattered in many different parts of Africa. And a lot of those areas are changing really rapidly, right? Areas of really high population growth, sometimes right in the middle of what we think of as biodiversity hotspots. So this is an important time to be doing this work and understanding this biodiversity before we lose quite a bit of it. So for the past 20 years, I've been doing field-based work across, especially Central Africa. So these are examples of trips that our lab has done, mostly in Central Africa, so Cameroon, Gabon, Angola, Congo, but sometimes some field work elsewhere as well. And the goal of that was, you know, I didn't set out to be a systematist. I set out to be interested in questions of morphological evolution, which I'll come to later in the talk. But it turned out trying to figure out if this weird trait and that weird trait are the same, we suddenly needed to know, are these the same species? Are these different species? How are these things related to each other? We really needed better sampling to build molecular phylogenies to be able to understand the relationships among these species. So a lot of this field work was focused on that and being able to understand speciation across this region. So let me give you a little context about why this is interesting. So diversification and speciation of African animals has actually kind of an interesting long history. I'm going to do, John is here. So John, this slide was not just for you, but here's a slide about birds. It's the only slide about birds I'll have. You can't see this. This came out in 1970. This is an incredible book. Has anybody ever seen this book in person? Other than John Bates? Nobody? Go to your library. This is actually 2 volumes. This is an amazing, amazing book, right? So this is 1970. It's a little small, but what I want you to point out here are these little dots, right? These are point localities, right, of birds. They tend to be grouped by species or groups of subspecies or groups of closely related species. The pitch of this was an atlas of speciation, thinking mostly about speciation being allopatric, right, of diversification across the landscape. But there's page after page after page after page after page of these maps of speciation of where these species are found. The thing that's kind of cool to think about, it's easy to lose sight of this on a map, but this is an incredible landscape, right? There's thousands of kilometers between the Ethiopian highlands and the Cape, right, of South Africa, right? So this atlas of speciation is on this really broad scale, and especially ornithologists have long been interested in, you know, it's really wild that you can be on a mountain in Cameroon and then there is no presence of this bird for 2,000 kilometers, and then here this bird is again over on this other mountain, right? And that really bothered people. That was true for birds, and of course, birds can disperse, right? They could fly between those places, and so there were a lot of argument about how these patterns came to be. You found the same patterns in trees. So Diego and I were talking about sky islands. The sky island version of Africa tends to be this Afromontane archipelago. This was an idea first put forward by Frank White. And the idea here was very similar, right? You have these species or closely related species that are found over these very large landscapes, but then nothing in between, right? So how did this pattern come to be? Was this dispersal from one place to another, you know, just skipping the whole area in between? Did these areas used to be connected to each other? Those ideas have bothered biologists working in Africa for a long time. So now we know a little bit more about the history of Africa to kind of give some ideas about how these patterns came to be. So one of them in a broad scale is that, you know, Africa in the Oligocene, early Miocene was is thought to be broadly forested. So that tropical forests were very widespread across a lot of Africa. And that beginning of the Miocene represented this big red band. There was increasing aridity in Africa, and that was where a lot of these more arid adapted grasslands started spreading, breaking up a lot of these widespread forests. So by the end of the Miocene, you have patches of forest, like in Central Africa, but a lot of the rest is a mosaic of woodlands and grasslands, and that's really the context in which our own species evolved, right? So during the Miocene, it seems like there's been this massive climatic transition from forests to more open areas, right? So we have a sense of that now from historical data. The other thing, within Central Africa and West Africa, there's long been thought to have been an oscillation of forests expanding and contracting. So as climate cooled and warmed, you had forests expand and contract over time, periodically. And those expansions and contractions have been believed to be sort of engines of diversification, bless you. And so some of that was based on actual historical data. So Jean-Marie spent a career sampling pollen across lakes across Central and West Africa to put together these maps of where forest was and wasn't through time, right? And so we have some historical record of this from pollen, but this is just the historical context, right? So an interesting thing now that we have molecular phylogenies, the fossil record, which I'll come back to, is poor for frogs, but now that we have molecular phylogenies, we can calibrate divergence times, we can begin seeing, whether the patterns of diversification kind of match these things in the Miocene, the Pleistocene. We do a lot of this work in my lab based on these endemic families that are found in sub-Saharan Africa. There's quite a few families of frogs that are found only in Africa. There are some remarkably cool frogs, so I'll just talk about a couple of them. These African frog families, we now know, have diversified over a pretty long period of time. We think a lot of these families were probably present in the Cretaceous, and that's based on not the fossil record, that's just based on time-calibrated molecular phylogenies. But we think they were probably there. A lot of the diversification among genera probably happened early in the Cenozoic, but probably, in the Miocene and Pleistocene is when we see a lot of divergences between genera and within sort of major branches within some of these widespread genera. So that's sort of at a broad level. This is of the group that's called the Afrobatrachia, which is an endemic lineage of African frogs. I'm going to give you a couple other more specific examples just to show you kind of how we think we know what we know. So the first one, so this family, these two families are widespread across all of sub-Saharan Africa. You'll notice that there's actually not that many tips in this phylogeny. There's probably 250, 300 species across this group. So this is very low resolution sampling and a look at diversification. So I'm going to tell you sort of a bigger scale example of purposefully sampling across an entire genus to kind of figure out how these lineages diversified. So this example is from Conrawa. So Conrawa Goliath is the largest frog species alive today, probably the largest frog species ever, actually. We don't know of any frog bigger, it turns out, than Conrawa Goliath. Their defining feature in this great field guide of frogs is that they're slippery and hard to catch, which I think is an amazing diagnostic trait. But there's a handful of Conrawa, and the thing that's interesting is their distribution. So here's a Conrawa day. You can see these little purple blobs. Many of these endemic families are pretty much found throughout sub-Saharan Africa. You'll notice maybe two patterns I'll just point out real quick. One is a lot of things are absent down here. So this area of Southern Africa is very arid and has been very arid for a long time. So a lot of these widespread families have very low or no diversity in Southern Africa. The other is that you'll see in an area that's not arid, a very low diversity of frogs, right? That's weird, right? Are there really no frogs there in this family? This is a remnant from the IUCN Red List, where we tended to have frog samples in museums from here and over here. And then this is the Congo River. So all these range maps just simply went along the north side of the Congo River, which for an individual species sounds reasonable. But then when you stack them up, it basically gives an appearance that, wait, there's no frogs in the middle of the Democratic Republic of Congo? That seems unlikely. And in fact, it is wrong. And so a lot of these range maps, I would say, if you're interested in Central African frogs, you have to take that with a grain of salt. But if you look at Conrawa, you'll see there's a little bit of West Africa, a little bit of Central Africa, and a little bit over here in the Ethiopian highlands. And that's actually a really weird distribution for frogs, having that disjunct distribution in Africa. Let me tell you about these frogs. So in Central Africa, where we've done a lot of work, and all these points on the maps are field samples from our collaborators' work, we find they're all cut off at the top. We have Conrawa Goliath, the biggest frog, Conrawa robusta, the second biggest frog, and Conrawa crassipes, a little frog. These are mostly, a lot of the diversity is here. Conrawa Goliath and robusta co-occur at the same place. They're both eaten by people, so we know a lot about these frogs because of that. In West Africa, we have two other species. Oh man, Aleni is on the left and Darui is on the right. These have kind of a funny disjunct distribution as well, because Darui is only found in this little area of eastern Ghana and Togo, and then Aleni has been found across several countries in West Africa. And then we get the truly one of the, this is Conrawa bakari. This is a species that was originally described from Eritrea, not a place known for its frog diversity, and then also is subsequently found through a lot of the Ethiopian highlands. This is a totally weird frog. It's probably one of the weirder frogs in Africa. They have these crazy sexually dimorphic heads. We really don't know what they're doing in the world, how they spend their time. There's probably some sort of male-male competition happening to explain the sexual dimorphism. But this is the pattern, right? We have these sort of disjunct things between West, Central, and East Africa. We can use time-calibrated molecular phylogenies based on a lot of nuclear and mitochondrial data to look at the relationships between these areas, as well as try to get a sense of the divergence times. So here, it's cut off at the top, but you'll see that Central and East Africa have kind of a weird relationship. It turns out that one of the species in East Africa is closely related to one of the species in Central Africa, but not the rest of them. It's an odd pattern. And then the West African ones form a distinct clade. But what's kind of more interesting for this story is that the divergence times are very old. They're really on this time scale of the Miocene. And so this separation between Central and East and West and Central seems to really be on the time scale of these Miocene forest fragmentations that we think from totally independent data have been happening. So it's kind of interesting to see within the genus these diversification patterns kind of mirroring that time scale. It's also resulted in a couple new species that have been described subsequently out of this. But it also, if you look within species or within groups of closely related species in Central Africa, we see a much shallower timescale of diversification. So because we've spent so much time sampling between Nigeria, Cameroon, Gabon, Angola, we now have a lot of sampling within species or across close related species, kind of what the bird speciation atlas was aspiring to. And we can look at the divergences among all these populations. And when we do that, we tend to find a lot of these divergences happening in the last 1 to 5 million years, sort of across the Pleistocene. And this was a paper from a while ago now, 2017, but there's almost like a cottage industry now of publishing these papers, of looking at population divergences across frogs in Central and West Africa, and they all kind of find the same pattern. This is a paper led by Raina Bell at the Cal Academy. This is just a couple almost randomly chosen hyperoleid tree frogs, where we have sampling across even the Congo Basin and the Atlantic coastal forest. And again, might be a little hard to see where you are. Most of these divergences are happening in the Pleistocene or Pleistocene-Pliocene, right? Sort of matching these patterns that we would expect if a lot of the divergences within species or between closely related species in Central Africa are being driven by these Pleistocene climatic fluctuations, right? What's next? Okay. One of the things that I don't have time to get into today, but is these refugia. Like, are these real? Do these really exist, these refugia? So a former grad student of mine, Greg Jongsma, we've done a lot of environmental niche modeling to sort of predict distributions back into the past and use that for kind of interrogating population genetics a little bit more now. So that's work that's in process and hopefully will get submitted sometime soon. But what I want to zoom out for is this, right? If you're looking across African frogs, like when I started this, we really had almost no idea about the timescales of evolution for African frogs. And I think what's kind of neat and has been emerging is that there's sort of 3 types of patterns that have been happening. So one is that, early on the stage was set, right? Where a lot of the African frogs have been African frogs since probably the Cretaceous, right? There's not been a lot of dispersal into or out of Africa, it turns out, except for like basically the lineage that led to bullfrogs. So that was set up in the Cretaceous. In the early Cenozoic and mid-Cenozoic, We have these sort of broad scale climatic changes that seem to be either separating genera or separating big groups of species within some of these genera. And then a lot of the speciation within these genera is really in the past few million years. And a lot of that seems to be on a regional scale happening from these climatic oscillations. And so now there's exceptions to all of this, but I think it's kind of in a broad way, kind of interesting to think about like this nested pattern of these diversification events happening. I started, as I alluded to, in paleontology. I've always been interested in fossils. So the last 10 years, we spent a lot more time working on fossil frogs. And really this is happenstance. It turns out that there's not a lot of fossil frogs that have been recovered from across Africa. So I've been working with Alfred Lemierre, who is currently a postdoc in the Royal Tyrrell Museum, was a PhD student in France. Together, we've been working through a lot of different projects going all the way from the Jurassic. So we have a paper that's back in review from these are the Tendaguru frogs, some of your paleontologists from Tanzania, this famous site that the Germans worked on for many years. There are some frogs from the Jurassic there. These are the first Jurassic frogs from Africa. And we have a lot of Cretaceous material we've been working through. And so my hope has really been to try to take these molecular phylogenies that make predictions about what we might find in the past and when we might find it and where we might find it, and sort of marrying those two things together. And so it takes a lot of work to sort through all these microfossils, but we've been slowly making progress on it. A lot of this is more deep time. One of the things I'm actually particularly excited about and have a fellowship application in right now is to go back and actually look at all the fossil frogs from the hominid sites in Ethiopia. All of these things, it kind of tends to be the only thing not described from these hominid sites. In almost every paper, they're just vaguely called frogs. So there's an enormous opportunity. It's like the one thing the paleoanthropologists have left. It turns out to be the frogs. So now that we have these digital libraries of CT scans of African frogs, like I'm going to hopefully go back to Addis and be armed with those data to identify a lot of these frogs from these hominid sites. And a reason that's kind of exciting is that frogs have these very specific habitat requirements, right? Some frogs breed only in these type of ponds or only in these type of streams. And so in a rough approximation, what type of frogs are found at these sites kind of inform us about the types of water availability that would have been at some of the hominid sites of our earliest ancestors, right? So I think that's exciting. I'll just say right now, like my time in field work in Africa has just been growingly limited with administration. It's hard to imagine getting to the field in Africa these days, but the fossil side is something I can do basically at any time. And since nobody's done it for the last 100 years, there's really not a rush to do it quickly. So I've been slowly working more on fossil frogs from Africa, sort of on the side. And that's an area for my lab right now. So I have a little bit of time. So I'm going to end on something completely different just for fun. So here's and now for something completely different. So frog biologists have cared about homoplazy, convergent evolution, a lot in the last 50 years. But it's also kind of boring, right? Like you've seen one frog, like Bob kind of nailed it. You've seen one frog, you've seen them all, right? You see these tree frogs evolve again and again and again. What about all the weird ones? I'm interested in the weird ones. One of the most interesting things in frogs that are unique traits that you actually only see evolve one time or two times tend to be male secondary sexual traits. They tend to characterize a whole clade, and it's weird, right? So there's so many things about frogs that evolve so many times. There seems to be almost nothing truly unique, but one of the rare areas where there's uniqueness are in these male traits. And so just as Stephen was just asking me, this is an antenna frog for you. There's the mustache toad. This is actually not completely unique, but a lot of male frogs that have weird spikes on them. There's the tailed frog, the only frog with a penis, that's a scaphus. And then there's my favorite frog ever, which is the hairy frog, which I won't come back and talk about. But there's all these weird male traits, right? And so I spent a lot of time worrying about a male trait in a group of frogs that I did my PhD on. So we're back to the group, the Afrobatrachia, and the two things I've circled here are what I'm going to talk about for the rest. These are little brown frogs. So as I think I claimed to somebody earlier, a real frog systematicist works on little brown frogs. That's like the cream of the crop in systematics, not these gaudy tree frogs. So this is my weird male trait. Just a weird finger. I've been bothered by this finger since I started grad school. I didn't pick to work on this because of the finger. I picked because of body size, all these other questions, but there was this finger And a paleontologist who was on my committee, Ferris Jenkins, always like refocused me on tell me about that finger. What's that finger doing? I was like, I don't know what the finger is doing. So here's what maybe the finger is doing, talk. So when I started this, I kind of started in the basic place because I had museum specimens, right? So I started focusing on trait diversity and evolution, and then tried to get into function, which was a mystery. So of course, this is a male trait that's kind of weird because, well, females have fingers, right? It's not like it's a unique trait, but it's unique in some species that it's relatively long for their body size, and we'll get into that quickly. One thing as a note is frogs only have 4 fingers. I am that parent that pauses all cartoon frog shows to count fingers on frogs. So frogs have 4 fingers, and the idea that we think is that they lost their first finger, right? And so this is actually important to this story. So the finger that's long is not their 3rd finger, but evolutionarily is their 4th finger, right? So that's what that long finger is digit 4. Now, as a grad student, I spent a lot of time measuring frog fingers and looking at frog fingers. And if any of you are doing your PhD and there's a moment where you're like, oh my God, is this really what I'm doing with myself? This is what I did with myself. I spent so much time measuring frog fingers. And an interesting pattern started emerging that nobody had ever talked about, right? So one is that on the inside of the 2nd finger and inside of the 3rd finger, inside of the 4th finger, there's spines. Weird. Most frogs don't have spines on their fingers. They have nuptial pads, right, on the hand, not things out on their other fingers. So that's kind of weird. Sometimes, and this actually, there's some species specificity to this. Sometimes they have spines on the wrong side of the finger, which is weird. I didn't know what to make of that, but I did a lot of that documentation. So, but it's going to be important, it turns out. As frogs grow, their fingers get longer. This might sound obvious, but it means that when the frogs are little, you have no idea if they're a male or a female, right? So in one of these groups, Cardioglossa, they have tadpoles. Arthroleptus are direct developers, so they start off as eggs and they hatch out as a little frog, right? So in either group, you can't tell a male from a female when they're little, right? So it only seems to be happening as they hit maturity. These fingers start growing rapidly. And that's true across like all the species we've looked at. And I spent a lot of time measuring like all the other groups of frogs related to these to see if maybe there was an inkling of this. And basically there's no evidence of any inkling of this in any of the other frogs. So seems unique in this family to these 2 genera. The reason I started working on these frogs originally was they had this wide range of body size, and I was interested in skeletal diversity and body size. So this group, the genus is kind of neat. So there's at least a 4 or so fold difference. So some small frogs like Arthroleptus kidogo, the little bit of a frog, is 14 millimeters long. You have some big ones that are 57, 60 millimeters. So you might think, well, maybe when the frogs get bigger, the fingers get relatively longer. You might think that, but you'd be wrong. That doesn't happen. All frogs have some dimorphism, which is kind of interesting. So like every species I've looked at least the male's finger might be a little bit longer. Sometimes it's almost not long at all, but it's still slightly longer. But it turns out to be a lot of little ones that have really long fingers, which is kind of interesting. And I have the numbers up here just because I have spent a career measuring little brown frogs at this point. So it makes me feel good to see how big the numbers are. You don't care about that. So another thing that's kind of interesting in evolutionary context is these long fingers have been lost several times. So it's a little bit like the story with peacock tail feathers, right? It's not unidirectional. They don't just keep getting longer and longer. Once you put in the phylogenetic context, you realize this trait has kind of gone back and forth, right? Sometimes it gets larger, sometimes it gets reduced. So that's happened in these frogs, which is kind of interesting. So we have at least 4, probably another case of reduction of this male trait. But we don't know why. Part of the reason we don't know why is I don't know what the hell they're doing with the finger, right? There's no explanation. So when I was a graduate student, the only explanation was this paragraph of text in a faunal account by Jean-Louis Amier, who died recently. I'm just going to read it to you in English. He said, I only witnessed once a rather extraordinary fight scene. 2 males standing on their hind legs stood entwined, the arms of each passing under the armpits of the other, the serrated 3rd finger vibrating ceaselessly against the back of their opponents. In this embrace, their bodies had an oblique position, blah, blah, blah. I don't know if you know what to do. I did not know what to do with that. And there's only one account. That's it. There's no other account. Until I got this video. This is the same species, actually, that Amier was talking about, which is really cool.
Stroking their long fingers in the back.
As Dan Mullen and John Weems as their females, they're not females, these are males,
The back in general.
they're wrestling. And they're wiping their finger against each other's back.
Did you see
I
that?
mean, it's
No.
useful in a way, because it's actually what Amie said they were doing.
Not useful because I still don't know what they're doing, but what's more interesting. Uh-oh.
Shit, this is serious combat.
Yeah.
Is there's more than one observation of this. This is a totally different part of the phylogeny of Arthroleptus. These are savanna frogs. They actually don't have that long of a third finger. And here they are in this combat.
Maybe that's why they.
Both of these species, as well as this third one, are direct developers. So they probably lay their eggs under soil. We don't really know what they're fighting over. The thing I thought was exciting in this video that Ed Basham sent me, there's often, these videos are all by male herpetologists, by the way, all of whom are like usually chanting fight at some point. This is a frog that actually doesn't have long fingers, but they're doing a similar behavior. And if we watch long enough, you'll actually see them do the same behavior of wiping their fingers basically against the other one. There he goes. And these fingers are actually very, this was one of my quote, reduced dimorphism ones. Which is
Hey, they're fighting.
So we don't know what they're doing. And I've spent a lot of time thinking about this, but there's pieces of it that the pattern suddenly made sense. Things that I had spent so much time counting and looking at under a microscope suddenly had some value. So one of them that I didn't tell you about is in their hips, right here. They all have spines too. That's also a weird trait in male frogs. You tend to not get these weird spines like this. That's exactly where their hands are in all of these videos, right? And I don't know if this is a defense against the other person's, I don't really know what to make of this, right? But it's kind of interesting because it seems integrated with what the behavior that they're doing. I was interested in if there was anything special about the fingers themselves. So we spent a lot of time CT scanning things, as some of you know. One of the things that's kind of interesting about this is how these hands and fingers are growing, which is kind of cool. So you can see the long finger here. You can see that there's a dense region and then a less dense region. So this is an X-ray image, right? So the brightest part is the most dense. So you can see this dense region that kind of looks like a normal finger bone, and then this sort of tube on the end. So that's kind of interesting. I'll come back to that. But you also see it in the hand bone, which nobody had ever said, oh, they have a relatively long hand. It's just been a long finger. You also see it sometimes in some of the other hand bones. You also see it in the radial ulna, which until I saw the x-rays, I never even realized that they had relatively long radial ulna. What's happening here? So what I think is happening is that these are cleared and stained specimens, a sort of special skeleton prep. This is the normal bone. This is the typical bone that a female has, the diaphysis, the shaft of the bone. Now, what I think is happening at the onset of sexual maturity, the distal piece, the end piece of that bone, which is typically cartilage and then ossifies, it grows rapidly. It hypertrophies and then ossifies and fuses to the other part. So you get this kind of funny composite element that is, you know, in some of these frogs, basically cartilaginous at the end, but it's like having a long tube stuck onto your regular finger bone, which is kind of weird. I thought, well, maybe this is why sometimes in collections, these fingers are broken. Maybe they're actually really fragile. Maybe this is why it's a costly trait to maintain and sometimes it gets lost. So I had a former grad student who's now at Trinity College as a professor do some finite element analysis. Now I'll just say this is not published. This was kind of this paper is published, but not this finger stuff is published. So first approximation, these fingers in ways that we tried to bend them digitally seemed as strong as their regular bone. So it doesn't seem like they're extra fragile. That's basically the punchline of this. I also was interested if there was any special other adaptations in the fingers. And so I had a visiting postdoc, Paulo Pinheiro, who really loved dissecting frog hands. And one thing we found is that the frogs in this genus have lost one of the bellies of muscle for their flexors. So basically when they flex, they automatically sort of go inwards, right? Because they've lost the flexor on the outside of the finger that would make it go like this. Instead, they're almost like they would, every time it flexes, it would rotate. It's kind of interesting. I was kind of, you know, me trying to wrap up these things to make sense of them. You know, here's a unique trait in frogs. I don't really know what the combat is about. We can have lots of discussion about that. I'm sure you have ideas. They're probably not as fragile and costly to maintain as I first thought. Maybe, maybe these bones are not that weak, even though they're kind of these weird composite elements. One of the things I found really kind of exciting about this is thinking about these as an androgen-driven trait, which makes sense, right? It's a male trait, it's secondary sexual. Probably most men in the room have a fourth finger that's longer than their second finger. This is the famed 2D/4D ratio, where it tends to be that this is an androgen-driven thing. Supposedly, I think there's some rule about if you're a werewolf, one of your fingers is longer. But that's really interesting, right? Because that's the fourth finger in these frogs is the one that's really long, right? And it seems like this is something that's happening at sexual maturity and it's growing long. And the reason this kind of made me excited was that this is a novel trait that seems to basically be evolving through a loss of a muscle and elaborating some basic developmental mechanisms that have been there all along, right? So while we tend to think about this as like a unique male trait in these frogs, and it is unique across frogs, seems like this is a trait that's evolved from some simplification and elaboration, right? It's not, a lot of times we argue about what novelty truly is, right? And this is kind of an interesting case of maybe loss and elaboration of something that's been there all along. And this is something that Dave Wade talked a lot about with, especially within bolitoglossine salamanders. And so I thought that was kind of fun to kind of connect, this novelty to that idea. I'm going to end with this. It's easy for us to sequence DNA now and CT scan things. I feel like we're all pretty good at that. But it turns out that natural history, like getting these videos, is just as hard as it was probably for Darwin. I guess impossible for Darwin to make a video, but at the same time, right, the natural history, trying to make sense of these weird traits is hard, right? You have to be in the field. There's really nothing that can replace that. And so for those of you that are especially students, I really encourage you to think about that, to spend time in the field, to do natural history, to catch those random videos that suddenly, I don't know if this totally solved things for me, but like they gave me those little glimmers of insight to begin making sense of traits and begin thinking about things in a new way. And that is impossible to do without that natural history data. And so even though these natural history papers are not looked upon highly by hiring committees and things like that, they tend to be low-level publications sometimes. They're super important for trying to make sense of these other patterns in biology. So if any of this has been exciting, you are excited to join the African amphibian community, please join us. When I started, I feel like there was like one or two of us in the US, and now we have a bunch more. This is a picture from the African Amphibian Working Group meeting that happened a year ago in South Africa. This is now a really collaborative, fun community of people from around the world. It's not just dominated by South Africans. There's people from across West, Central, East Africa, US, Europe, even South America in this picture. So if you're interested in these things, we'd be happy to have you join us. And with that, I will end. Thank you. Well, all right.
Yeah, Dave, thank you so much we have a lot of questions. Tom, do you want to do it first?
Hey, Dave, thanks very much for coming. Quick question. When you saw these frogs in combat, did you notice any, from those spiny figures, did you notice any abrasions on the skin of the opponent?
That's a great question. To our knowledge, I've never seen any in these frogs. I've never seen that. They're kind of dainty little spines, so I don't know that they would really result in that. But that is common in a lot of frogs that do fight. They have bigger, bonier spikes, and you can definitely find like open wounds and scars and things like that. But not in these frogs, no. It's more of a tickle. Maybe. That was a hypothesis someone threw out at me, was they were tickling each other to release sperm, which sounds awesome. I don't know. Stephanie?
Okay, so with the long finger thing, how does it affect locomotion at all? Do they like trip? Like what's the deal with it?
Yeah. I don't know.
Oh, you don't know?
To my
Okay.
knowledge, no. There were thoughts early on that it was like used for like grappling. Like, males would use it somehow to climb. Never seen a male frog do that. There were also ideas that somehow it was used in plexus. but like there are images of these in Plexus and they basically, like, they don't use their fingers at all. They're holding them just hand to hand, you know?
Personally haven't seen one like trip over something.
No. I mean, I'll admit to being that graduate student who would catch male frogs and like put them in a jar and be like,
Yeah.
will something happen? Come on, do something. No, I've never been able to actually see it myself, unfortunately. But you know, they live in leaf litter, almost all these frogs. You know, it's hard to imagine that being a huge locomotion problem for them. I mean, they're pretty cryptic. They live under leaves, you know. I think this is where all the action is when they start doing things. Most of the time they just sit around under leaves is my general impression of them. Yeah. With their long fingers frequently. Yeah. Yeah. Oh, yeah.
All right, two questions. Is there a gland somewhere located, or is it just like a spiny region on like that back portion that's being rubbed? And then if the answer to that is no, then I'm curious if there is some kind of cranial nerve pathway that might be passing through that area. Like, are they trying to like hit a pressure point or something? I'm really curious. Like, how are these fights ending? Because none of those videos showed the ends of the fights. Like, what's the end result here?
Yeah, awesome. Glands, not in the macro gland sense. So in a lot of frogs, like in a toad where you see its parotid gland, you're like, okay, it has a big gland there. They don't have anything like that. To my knowledge, there's never been histology of that region to see like, well, maybe there's something under it. That's never been done. How do they end? In fact, I only have one video of it ending in some obvious way. And it's actually the first video. And what it shows is that at the very end of it, one frog is on top of the other, and the frog on the bottom like does this weird, awkward like squeak, and then leaves. Like it's a release, like it's been dominated, and now it's like getting out of there. I don't know, you know. And what's interesting, a lot of these frogs is that so a lot of frogs when they fight over stuff, females, access to calling sites, access to egg laying sites. These are frogs that lay their eggs in the soil. There's no females around in any of these videos. So like, I don't know what that means, you know, or are they, it's hard to know what exactly they're fighting over. I do wonder about calling sites. But that's something that really is going to require like real work in the field to kind of figure out like, you know, even how males space themselves out to call. It's kind of never been looked at. Yeah. But it's, yeah. So I don't know. It's basically, yeah.
I just had two development questions. I'm over here. And one ecology question, a development question. I mean, frogs, as part of their bio plan, get rid of the neck. It's so unbelievable because even the salamanders keep some movement there. How does that happen? It's never been reversed. Do you see anything before it disappears? Second genetic question, they've added a limb length to the hind limb. They took a tarsal, made a long bone out of it. Is anything more known about the genetics of it? Because it sort of almost resembles like taking a fin and getting your first long bones. And then the ecology question is, I've never seen such adherence to these continents for animals that are so water adapted. They seem just incapable of making that transition out into the ocean. I don't know why. I don't know why there's not more dispersal for animals that have such unbelievable aquatic capabilities. So those are all impossible questions.
All right. Neck. No frogs have a neck. Most frogs have 8 vertebrae between their head and their sacrum. There are frogs that have less than that. It tends to be because some of the vertebrae are fused together. So there's a lot of little frogs where the first 2 vertebrae are fused together. Triatobatrachus, which is the only stem frog that has enough of a, you know, I'm struggling because I'm trying to think of how the head is connected there. There's more vertebrae, but I don't know that there's ever been a study to guess if there's quote cervical vertebrae or thoracic vertebrae, which is interesting actually to know. So not a lot known about that. Your next question was about.
It doesn't appear and disappear. It's just deleted from.
No, there's no, and in fact, the only trend of vertebral columns is reduction. And frogs are remarkably conserved in that. You asked a question about overwater dispersal.
And then a question about the parcel to
Oh, yeah. Oh, great. All questions, yeah. So overwater dispersal does happen in frogs. So don't let anybody tell you otherwise. However, it's very limited. It tends to seem to happen in cases where there's what has been referred to as freshwater lensing, where you have large surges of water come out of river systems that are meaning that the ocean water tends to be much more fresh water on the surface. And so these frogs may be able to persist and disperse. So for instance, that's how we think frogs probably and even Sicilians managed to get to Sao Tome and Principe, which are well off the coast in the Gulf of Guinea. There's volcanic islands. There's never been any land connections there. It's also probably how frogs managed to get into some of Oceania. There's frogs out there. There's no other way they could have ever gotten there other than raft there. But it still is pretty uncommon, and there are frogs that are more saltwater tolerant. So whether or not that's a clade-based thing is not known. The last one, tarsals. Yeah, so frogs, all living frogs and all proto-frogs have their radius and ulna and their tibia and fibula fused together. There's actually no variation in that at all. It's never been reversed. Sometimes early in development, you can find these as two bones, but they always fuse. Interestingly, then what Paul's referring to is their tarsal bones are also long. And what's kind of cool there is that there are examples of frogs where the tarsals have also fused and are effectively a third single bone, right? Which is really wild, right? And there's one family of frogs where there's actually variation in that trait, which suggests it's an interesting group to like kind of get into like, how does that even happen in the first place, right? That's Centralinix glass frogs, if some of you know that. It's a whole reason glass frog, nobody cares about glass frog tarsal bones, but it is probably one of the coolest things about glass frogs is there's variation in that fusion for that third element, yeah. But I've never seen a developmental study of tarsals.
It's stunning because it's sort of like, that would be your tetrapod, thing.
Yeah, no, it's a great question.
We've got one last question.
Oh, yeah.
Can I ask? I work with insects, and when I get a nice molecular phylogeny, usually we see those Gondwana splits, and then the only fossil that exists is Baltic amber, something like this. So how has this, like, now that you're looking more into frog fossils, what have they revealed about? Have they revealed anything new about the history you have based on phylogenies, or
Yeah.
is it pretty much the same story?
That's a great question. Frog fossils have proven very difficult to work, right? So on the one hand, there's an incredible amount of frog fossils. The disappointing part is they're all fragmentary and distal parts of a bone, right? While we do publish papers on a distal humerus only, it's not a lot you can say based on that. On the flip side, some of the best fossils that we have are so crushed flat that it can be very hard to pull characters out of them, right? The other part is frogs don't have a lot of bones. So a lot of the big phylogenies that have been built for like squamate reptiles, for instance, their character matrices can be hundreds of characters, right? And hundreds of taxa. You know, most of the morphological phylogenies for frogs are like, 20 by 50 matrices. There's like almost no information, right? So a challenge for us now is really building those character sets to do this. There are still, however, it's not all gloom and doom. There are still frogs that we feel pretty confident, like, okay, this is the oldest member of this group or that group, right? So then that's how we end up calibrating these phylogenies. But trying to figure out how to make better sense of the frog fossil record is something I'm super interested in. Some of you know Mena Jones, who's a grad student in Chicago, who's working on fossil frogs. I had a fossil frog PhD student and now postdoc. A lot of what we've been spending time on is trying to think like what more we can get out of the fossils and how that can actually inform our understanding of living diversity, And it's hard to get characters out of fossils. Because of the nature of a lot of the fossils, boy, it's hard to even like, what would you do with like a distal insect leg? It's like you could look at cuticle and be like, well, maybe this one group has this weird cuticle, like that's about it, right? So we've been thinking, in the mammal and bird, especially in the mammal world, dinosaur, archosaur worlds kind of like this. They've spent so much time thinking about what they can eke out of bits and pieces. A great example is like mammalian teeth, right? Like how many papers are written based on mammalian teeth as being informative of something, which they are. So we've been really thinking a lot about, well, what's the frog equivalent of that? Teeth not gonna help us, unfortunately, for frogs. But we have been thinking a lot about, inferring body size or inferring locomotor modes and things like that. So even if we can't put a fossil in a phylogeny, you could characterize a fauna at a time as having this distribution of body sizes and this distribution of, habitat types and things like that. It's not helping for calibrating a phylogeny, but is helpful for thinking about, like, through an extinction event, did you lose all the big frogs? Did you lose all the tree frogs, right? Like, I mean, that's kind of how we've been trying to approach that question. I mean, sometimes we get amazing fossils and you can code them in a matrix and put them in a tree, but unfortunately, that's a limited part of the fossil record for us. Great question. All right, thank you very much. Appreciate it.